Welcome to The Explainer. Let's jump right in. Today, we're exploring a highly complex, incredibly powerful, and, honestly, just an endlessly fascinating piece of scientific technology. We're talking about gas chromatography-mass spectrometry, or GC-MS for short. We're going to take a journey into the microscopic world, where we take complex mixtures of chemicals, break them down into their absolute most fundamental parts, and figure out exactly what they are and just how much of them is actually there.
To give you an idea of just how powerful this is, let's go back to the 1970s. When the Viking landers touched down on Mars, scientists didn't just want pictures, right? They wanted to know if there were organic molecules hiding in the Martian soil. And the exact fundamental technology they used for that extraterrestrial search? It's the very same technology forensic scientists use today to solve crimes, and sports agencies use to catch athletes doping. It is literally the gold standard for chemical analysis.
Okay, so let's dive into this. What exactly is GC-MS? Well, it's a combined method. First, you have gas chromatography, or the GC part, which gives us the ability to separate a really complex mixture. Then, you tack on mass spectrometry, the MS part, which gives us the power to detect and identify those separated parts. Put them together, and they allow for both qualitative analysis, meaning, what is this compound, and quantitative analysis, meaning exactly how much of it do we have. Here is our roadmap for today. First, the ultimate chemical detective. Second, GC, sorting the mixture. Third, MS, identifying the molecules. Fourth, real-world applications. And fifth, limitations and innovations.
Starting with Section 1, the ultimate chemical detective. Let's look at the journey of a molecule.
To really understand how this machine works, we're going to follow the physical journey a single sample takes from start to finish. There are 4 major steps here. First, it gets vaporized, literally turned into a gas. Second, it gets separated in the GC phase. Third, it gets ionized, which basically means it gets zapped into charged fragments in the MS phase. And fourth, those fragments are finally detected and measured. It's just a beautifully logical process.
Moving to Section 2, GC, sorting the mixture. This is the sorter phase.
So, let's look at the first half of this machine, the gas chromatographer. Here's what's physically happening inside this sorter. The sample is injected into a heated GC inlet, where it is instantly vaporized. Now that it's a gas, well, it needs to move. So an inert carrier gas, usually something like helium or nitrogen, pushes that vaporized sample right into the GC column. Now this column is a long, super thin capillary tube that's coiled up inside an oven, and coating the inside of this tube is something called the stationary phase, a fixed material that the sample constantly interacts with as it gets pushed through. Now, as the mixture travels through that column, different molecules interact differently with that stationary phase. Think of it kind of like walking through a really crowded party. If you don't know anyone there, you have weak interactions, right? So you zip right through the room and move super fast to the exit. You elute first. But if you know everybody, you have stronger interactions. You keep stopping to chat, you get bogged down, and you move slower, which results in a longer retention time. Because of these different speeds, the complex mixture separates out in a literal microscopic race to the finish line.
But, that brings us to a massive question. We've separated the mixture into its individual parts, but how do we know what's actually there? The GC only sorts them. It doesn't actually tell us what they are.
Which brings us to Section 3, MS, identifying the molecules. This is the identifier phase. As those separated molecules exit the GC, they travel through a blazing hot transfer line, called the interface, directly into the mass spectrometer. It has to be kept incredibly hot to ensure the sample doesn't degrade back into a liquid. Once inside, the very first thing we have to do is ionize them. We have 2 main ways to pull this off. First is electron ionization, or EI. This is a hard method. We aggressively bombard the sample with high-energy electrons, causing the molecules to just smash apart into fragmented ions. Alternatively, we have chemical ionization, or CI. This is a soft method. Instead of smashing them directly, we use a reagent gas to gently transfer an electrical charge to the sample molecules, resulting in way less fragmentation. EI is for sure the more common method, but both serve to give our molecules a specific electrical charge.
Okay, so now that we have a bunch of charged fragments flying around, we need to make sense of them. This is the job of the mass analyzer. It's a component that separates these newly created ions based on their mass-to-charge ratio. Think of it as sorting these microscopic fragments by exactly how heavy they are and exactly how much electric charge they carry.
And there are several wild mechanisms scientists use to do this sorting. A single or triple quadrupole uses parallel rods as mass filters to actively block or allow certain fragments through. Time of flight shoots ions down a vacuum tube to literally measure their speed, because, you guessed it, lighter ions travel faster. A magnetic sector uses a strong magnetic field to physically deflect them based on their weight. And an ion trap uses electric fields to catch them in a temporary orbit. The mechanisms vary wildly, but the goal is always exactly the same: isolate those fragments based on their mass-to-charge ratio.
Finally, our molecule's physical journey ends at the detector. It measures the intensity of every single ion that hits it and converts those physical hits into electrical signals. The computer then takes those signals and creates a unique mass spectrum, basically a molecular fingerprint. When you combine all these signals across the entire time it took the sample to run, you get a graph called a total ion chromatogram, or TIC. Each peak on that graph represents a different compound, and here's the cool part: the area under that peak tells you exactly how much of it is in your sample.
Let's look at Section 4, Real-World Applications and Impact, why all this science actually matters. Now that we understand the heavy science of vaporizing, sorting, fragmenting, and detecting, let's anchor this in reality. The sheer versatility of this technology is staggering. In environmental monitoring, it tracks pesticides. In forensics, it analyzes biological samples from crime scenes to detect illicit drugs like cocaine or poisons. In anti-doping labs, it catches cheating athletes by detecting anabolic steroids. Security agencies use it at checkpoints to literally sniff out explosives. And in medicine, it's used for newborn screening to catch congenital metabolic diseases super early. The exact same process that searches for life in space, keeps our food safe, and solves complex crimes right here on Earth.
Finally, Section 5, Limitations and Future Innovations, the Challenges and What's Coming Next.
Of course, no technology is perfect, right? While GCMS is incredibly powerful, it definitely has some hurdles. First, these machines are exceptionally expensive to buy, operate, and maintain. Second, you can't just throw a raw sample in. Sample preparation, stuff like extracting, diluting, and cleaning, is really complex and time-consuming. Third, and perhaps most importantly, the analyte absolutely must be volatile, meaning able to become a gas, and thermally stable. If a molecule degrades when heated in that oven, it simply cannot survive the journey through the machine. And finally, interpreting those massive datasets requires highly skilled personnel.
Because these machines are so sensitive, personnel have to troubleshoot constantly. They have to translate visual errors on a computer screen back to physical problems in the machine. See a poor peak shape on the graph? That could mean a physical gas leak, or maybe the user just overloaded the sample. An unstable baseline? That's likely contamination, which you can usually solve by using higher purity carrier gases. What about no peaks at all? Well, you probably have a physically blocked syringe or a straight-up broken column. It requires a really deep understanding of both chemistry and engineering to keep these machines running smoothly.
But the industry is rapidly innovating to overcome these exact challenges. High-resolution mass spectrometry is pushing mass accuracy to extremes. We are also seeing the development of micro-GC systems. Instead of a giant machine taking up a whole bench in a lab, they are shrinking this technology into portable systems for on-site analysis. They are also building in smart, automated systems to reduce manual labor, and designing them to be way more sustainable, using far less energy and precious carrier gases like helium.
Which leaves us with this final thought. We've seen how GCMS takes the complex physical mixture, races it through a heated tube, smashes it into charged fragments, and reads its unique digital fingerprint. So as this technology gets smaller, smarter, and faster, what unseen molecules will we discover next in our world? Or beyond it? Thanks so much for joining me on this explainer. I hope you walk away with a newfound appreciation for the ultimate chemical detective.